are the first time that we started adding in nitrogen. So we have the full HONC, okay, so HONC, which if we had carbohydrates, we had carbons and hydration OH. When we had fats, we had long carbon chains with a carboxyl group and a methyl group. So again, C's, very little O, but H. So the very first time that we are introducing the full HONC here. So when we studied our first chemistry video that we watched, we discovered that like most of life is composed of HONC. So that's where I'm getting the HONC from. Okay, so the NH2 is called an amine group. And so that is our functional group. And when we connect that with a carboxyl group, and there's like a carbon in here with like some other stuff attached to it. This is the amino acid. So an amino acid is the building block of proteins.
Is that an I?
COOH?
We get C. Up there's an R. And then
okay, so it's just like I was just kind of like saying there's something down there, but the R group is like a generic side chain. So I don't expect you to memorize that really, other than we understand that that functional building unit is an amino acid. What we are going to focus on is that we link together these amino acids. So amino acids okay, so amino acids link together in a polypeptide chain. So poly means many. So where does this peptide come from? These bonds in between the amino acids are called peptide bonds. So all of these amino acids are going to be linked together in a certain order with peptide bonds. So proteins can be called polypeptides.
in a certain bond?
And it is a peptide bond.
Protein
So the whole thing is a protein, right? Or a polypeptide. These bonds in between the amino acids are peptide bonds.
Oh,
And they link together the amino acids. Is that good? Give it a minute. An amine group is NH2. So,
always
yeah.
just NH2.
Yeah.
NH2.
It's a functional group, just like the carboxyl group. You still need that?
The stuff on the bottom. I can't see it down on the bottom. Very bottom.
Amino acids linked together in
Okay. So now we want to kind of like go back to like, we know what protein is, right? So what are some things that we know are made of protein? Like, yeah. So when we're, you guys are in sync. Okay. So a steak.
Fish. Oh, I guess so.
Yeah, so a steak, fish.
Eggs.
I will use boneless skinless chicken
So just
chicken breast. Well, the reason that I will, why would I do that? Use a boneless, skinless chicken breast
because
to
the
represent
bone
protein.
and the skin is not meat.
It's not protein directly, right? So, like, if I have the skin, it would be really fatty, so people might think it's a lipid. The egg white is protein, but the egg yolk is cholesterol primarily, so that would be a lipid. So I tried to keep it very straightforward, show you a steak, like the meaty bit, anything fatty. Nuts do have a lot of healthy fats in them, but so I probably wouldn't example, but they are good sources of protein as well. Hair. Do we know our hair and nails, fingernails, are a protein called keratin? Something that, because you've heard that your hair is dead, right? So it's just a dead protein hanging off. Okay, well now we know. So hair and nails are made out of keratin. It's a protein. Okay, so these are the things that I want you to know. If I gave you a picture of a steak or something like that. But we also think of our own muscles, so I'm going to be just terrible at drawing a skeleton, moving a muscle. But, you know, like if we're like flexing, we're going to think of our own muscles, and that be protein, right? So, yeah, muscles, I think we can relate to that's basically our steaks. It's our own meat steaks. But proteins are really much more diverse than that. So these are going to be our solid real life examples. But then we want to be able to also know their different layers of structure. So if I draw just the amino acids linked together, and they usually do draw them like a string of beads linked together with these peptide bonds. That is primary structure. So if I give you a picture like that, I do want you to be able to label it protein. The next layer of structure and organization, they call them, and maybe we should put 2 layers in there, that they're like organ folded, but they're called beta sheets, or folded sheets, pleated sheets. Then we have an alpha helix. So this is the secondary structure. So it's basically saying that this primary string of beads is going to now be attracted and form bonds that fold it into a specific shape following one of 2 patterns, either the beta sheet or the alpha helix, which is just a corkscrew. So this is secondary.
They're both protein.
Yes.
All 3 of them are ways to represent.
Yes. So this is secondary structure of protein. So if we see beta sheets or alpha helixes,
It's protein.
it's protein. Yep.
And what
Oh, sorry. I didn't write it out. It's alpha helix.
Is there different stages to get there?
So these
Or is
kind
this
of
primary,
are,
Or is it just all the same, just different ways?
well, okay. So like these are all representation models of protein, but the thing that our ribosomes would be putting together, so we will learn more in depth about protein synthesis, but our DNA actually gives us the recipe for the order of how we want our amino acids to be glued together, right? So we have that DNA script inside of us for everything except the proteins that we have to eat. So those are called essential nutrients, but there are some proteins we don't build with our DNA. We have to eat them. But for the most part, we have the recipe inside our DNA, and it's telling you the order of the amino acids. That is what the ribosome does. But then we're going to travel outside of the nucleus. Well, we're already outside the nucleus if we're in this order. But there will be other molecules or places that that protein will travel to be folded into its specific structure.
Okay.
So this primary structure is like the actual beginning construction of the protein.
Okay.
And then,
into
yeah,
okay.
yeah, then the chemical bonding attractions will form these kind of naturally. And then we get to tertiary. And the tertiary structure is like something we might be kind of used to seeing just as like a random globular shape. And I guess I shouldn't have made it so globular. Those alpha helixes and beta sheets will be in there. So this is the third layer. So this is a sort of a globular form. Now, this would be considered like one strand of DNA or one section of DNA, one gene coded for this protein. If we have multiple genes coding and working together to perform like a similar function, it would be quaternary. So we want to realize like the next layer up is if we had a string of DNA that coded for a section of protein, and that to be a fully functional protein needs to bond with another gene section of protein. Then we have quaternary structure. So it's multiple genes were involved. So I'm going to draw like kind of a famous one. And yeah, this is a very rough drawing. Like yeah, so it has 4 strands in there. And each one of these will have an inside hemigroup. And it's not showing up very well, but we'll try to do our best to show it. And inside the hemigroup, there will be an iron. So if we blow that up, it was a hemigroup and an iron. Okay, so that iron is going to attach to an oxygen. So that being said, so we have 4 proteins all involved. There are 4 hemigroups and 4 irons all holding on to oxygen. Do you know that protein? Yeah, have you ever heard of that protein that carries oxygen inside our bodies? That is an organ. That's a good one. So lungs is a good choice. But it is hemoglobin. So hemoglobin's like one of our famous ones that probably will be an example. And there's another 2-stranded protein that's pretty famous. So I'll just draw like a string and then another string. Well, I didn't put in the peptide bonds, but it is 2 strands working together, but it's insulin. So we can look that up so that we can get it more accurate. Yeah, so here's some like really pretty pictures. So again, our DNA is coding for the order. So if I give you this string beads, that is primary structure. If we're folded and we have our alpha helixes here, right, then if it's just the alpha helixes, that would be secondary structure. But since this is one colored strand representing this protein here, and this is a totally different colored strand representing this protein here, then is this tertiary or is that quaternary? Quaternary. So it's not just folded into its single globular shape. It has the two strands working together to perform that function, functional protein. So insulin is a very common protein that we all need. Do you know what its function is? What does insulin do in our body? Yeah, so it regulates our blood sugar, so it makes the blood sugar go into our muscles. So when somebody's insulin resistant, they are not able to get the sugar actually into their muscle even if they have insulin. So lifting weights is good for us. Okay, so just really neat structures like this. If you see anything beautiful, you will just label it protein. So I won't give you just chemical structures that don't show definition. I'll give you the cool, curly, cute ones.
We know that is a protein.
Yes. So hopefully.
Colors equals protein.
Yes, and multicolors means that it's quaternary. So these structures operate like within a fixed range of pH, temperature. What else would impact it? Like all kinds of things. So if it's exposed to a different electrical current, say you got shocked by an electrical outlet or struck by lightning, it would fry all your proteins, right? So many things change the shape of a protein, and that can result in bad things for us. Okay, so I'm going to stop share and pause, not that record.